Well, good morning and welcome to the first morning session of the Conference of Darwin in the 21st Century. Thank you, Greg, for those beautiful opening statements that really helped provide the context of the university as well as societal context for this particular conference. So today's session this morning is on current issues of evolutionary theory. And when we were discussing what topics we could choose for this particular session, there's a lot of different things that came to mind. Because there's really a lot of active research today on evolutionary issues that would deserve attention. But here, what we've done is assemble three very distinguished speakers who all have made significant and lasting contributions to our understanding of a very fundamental issue in evolutionary biology, and that issue is the evolution of biological complexity. So each speaker has actually made contributions in this regard, but their insights are from rather different perspectives. So I think overall, we're going to be talking on a similar theme, but very different perspectives with respect to that theme. So first, we'll explore the evolution of biological complexity on the level of the genome, and look at molecular mechanisms that have come into play, and then we'll incorporate how developmental biology and developmental mechanisms will actually directly influence the range of phenotypic outcomes that are possible, placing limits on the evolution of form. And then finally, we'll explore the insights that can be gained by taking the long view, and I mean the very long view, as offered to us through the examination of Cambrian fossils that represent our earliest metazoan ancestors from 500 million years ago. So through these three talks, I'm hoping that we'll gain a much expanded understanding of evolutionary processes that go beyond Darwin's original formulation of evolution by natural selection 150 years ago. So before I introduce the speakers, I just want to give you a brief overview of how this session is going to run. The concept is that each speaker will be given a chance to speak, and there will be some questions that follow each talk. And I would like to try to keep those questions rather informational or short and save some of the more in-depth kinds of discussion questions that are possible to the end, where all three speakers, again, will be available to actually form a sort of group discussion. So that's the idea of how we're hoping to do this, and I hope that we'll be able to follow through on that particular plan. In this way, I think we'll be able to get more at the synthesis between the three talks rather than just having each talk be presented in isolation. So our first speaker is Michael Lynch. Michael Lynch is a distinguished professor in the Department of Biology at Indiana University in Bloomington. Michael is also an associated investigator at Indiana University Center for Genomics and Bioinformatics. Currently, he's the president of the Society of Molecular Biology and Evolution, and he's also been president at the American Genetics Association and the Society for the Study of Evolution. He's a fellow of the National Academy of Sciences. He's also a fellow at the American Academy of Arts and Sciences, and also the American Association for the Advancement of Sciences. He has published 4 books covering a range of topics, from quantitative genetics to endangered species to genome architecture, and he's also published close to 200 articles in leading scientific journals. Without any further ado, I'd like to introduce Michael Lynch, who will be speaking on evolution by non-adaptive mechanisms.
Thanks very much, Hope. I actually was an undergraduate at St. Bonaventure University. It's always daunting to come to the mothership here, the big Catholic University, Notre Dame. You've been staring at this slide for a while. This is actually the first slide in my talk. And Hope, you're going to probably have to stop me at some time. I think we're starting about 10 minutes late, so I've lost track of when to stop. But I guess when you start waving your arms, I'll know. So all of you have walked into bookstores before and find daunting piles of books on evolution. And it's really remarkable that the number of people who regard themselves as experts in the field of evolution. Many of these books are written by people entirely outside the field of biology. Others are in biology but have made no attempt to really learn the basic principles of evolutionary biology. Some of these are by evolutionary biologists who really haven't always dug too deeply in the discoveries that we've made in evolutionary biology in the past 50 years. Now, there is this famous book here on the left that we're all celebrating. There's something like 1,000 Darwin festivals going on this year. It's quite remarkable. And most of you think of Darwin as the father of natural selection. And of course, he promoted natural selection as a major evolutionary force more than anyone. The idea that in start with him. What I think is most important about Darwin is that he was the big integrator, in my opinion. He integrated various fields of not just the biological sciences, but the geological sciences and so on, like no one had ever done before. And it was interesting to hear the dean emphasize integration of the sciences from the context of evolutionary biology. I think Darwin would be flabbergasted as to what's going on today in terms of discussions we're having about evolutionary biology, because it certainly is not integrative. It's as though nothing has happened since Darwin wrote his famous book. So that's my first bit of pontification. And the problem is this, that for many people, not just on the street, but biologists even, with PhDs in real biology departments, evolution is nothing but natural selection. And that makes it easy to think it's very easy to be an evolutionary biologist. You can teach natural selection to a first grader, and they'll comprehend it in a matter of an hour or so. It's easy to understand natural selection. The problem here is that natural selection is just one of 4 primary evolutionary forces, and we've known this for a very, very long period of time, for a good century. The remaining forces of evolution are 1, mutation. Mutation creates the variation upon which natural selection acts. Mutation is not a random process. I'll talk about this in quite detail. We've known that for a very long period of time as well. Recombination reassorts molecular variation within and among chromosomes. It also is not a random process. And there is this process here called random genetic drift that I'll be talking about, which is indeed a random process. And this is due to the role of chance in evolution. Hopefully you'll come to appreciate what random genetic drift does. There's a lot of miscomprehension about genetic drift. For many people on the street, well, I guess people on the street don't think about random genetic drift all that much, come to think of it. But those who do, outside of the field of evolution, think drift is sort of an ill-defined process that just creates background noise and alters the probability of different outcomes, but not the general trajectory of evolution. And that's certainly not true. In fact, just as much as natural selection is a guiding hand in evolution, so is random genetic drift. And what I really want to emphasize is we know more about random genetic drift as a process in a quantitative sense than any of these other forces of evolution. Drift alters the course of evolution. Drift is a sort of noise on the filter, or a filter for the eyes of natural selection. As population size is decreased, the role of chance becomes more important in evolution. That makes it harder for natural selection to move things forward. It makes it easier for mildly deleterious mutations to accumulate in populations, and it makes it more difficult to advance adaptive mutations. We live in an incredible time to be evolutionary biologists. We have all resources at hand to understand evolutionary biology due to our ability now to sequence genomes at a very high rate and at an increasingly economic way. Soon we'll have 1,000 human genomes, for example. In 5 years, we'll probably have 10,000 or so on. And by then, you'll all be able to get your own genome sequenced with a fairly good chunk of pocket change, let's say. If we want to understand evolution, there's 3 minimum requirements. First of all, if we want to propose a mechanism for evolution, it needs to be consistent with basic population genetic principles. Evolution is a population genetic process, and if an idea about evolution is inconsistent with population genetics, it's a non-starter. This is the big hole in evolutionary biology today, what goes on inside the cell. We've thought a lot about what goes on outside of organisms, going back to Darwin, but we really need to start thinking about what goes on inside cells in terms of molecules, molecular interactions, and so on. Just as a community ecologist needs to understand what species do in an ecosystem to understand that ecosystem, the role of molecules in cells really puts constraints on what can happen in evolution. Now, what we've done very well for a very long period of time is this, to think about the external environment and how that imposes evolutionary changes on populations. By this, I mean natural selection. We have thousands of very well-documented examples of natural selection in action. But as I said, for many biologists, all of evolution is due to natural selection. And I've likened this idea that every aspect of biodiversity can be explained by natural selection as not being very different than the intelligent design view of the world. In the intelligent design view of the world, you're just looking at some ill-defined force explaining all of biodiversity. And here, if you simply believe that natural selection explains anything, it's just this ill-defined force that we don't always have ecological explanations for. And I'm not trying to say that natural selection isn't an important evolutionary force. What I am trying to say is the standards in evolutionary biology should be set no lower than in any other field of science. And if we really believe natural selection is the explanation for some aspect of biodiversity, we ought to be able to reject the null hypothesis. There ought to be an alternative null hypothesis. And the null hypothesis I'll be talking about today with respect to genome evolution and gene structure evolution is an evolutionary hypothesis in which all the driving forces are non-adaptive in nature. And what we'll see is it's very difficult to explain the evolution of genome architecture and gene structure without invoking an absence of natural selection as a guiding force. Now, we've learned a lot about genome sequencing over the past few years, and one of the things we now have a very good handle on is the relationship between gene number in organisms and the complexity of organisms. And the bottom line is there's essentially no relationship between the number of protein coding genes and the complexity of organisms. So bacteria have up to 7,000 protein coding genes in them, and then we get up into this big eukaryotic group, including the big multicellular things that you look at every day. The human genome has about 22,000 protein coding genes. The pufferfish here has about 50,000 genes, as do most other fish. This giant monstrous cell, paramecium, which we work on, has about 45,000 genes in it. Now, most of you would argue that the human phenotype is much more complex than the phenotype of paramecium, although the paramecium aren't talking about this. But the main point here is you can make a much more complex organism, this cell, with many, many fewer proteins. What is going on as we go from these minuscule microbes here to us big multicellular species is changes in other aspects of the genome. Not gene number, but the amount of DNA residing between genes and even within genes, as I'll point out now. This is a set of 3 plots of the scaling of genome content with genome size in various organisms, going down here from these viruses all the way up to land plants and multicellular species. I've broken the genome just up into 3 components here. Here's the coding DNA as a function of genome size. For viruses and bacteria and the smallest eukaryotes, almost all the genome is coding DNA for proteins, about 95% plus, and then it levels off, and that's because, as I said, there's essentially, eventually, in eukaryotic species, no relationship between gene number and the complexity of organisms. The human genome's out here. Only about 1.5% of our genome is involved in coding proteins. What's going on then as you grow larger genomes and more complex organisms is a sudden transition, although it's a smooth transition, from almost all coding DNA to almost all intergenic DNA or intronic DNA, which are inserts inside our genes that I'll mention in a second. What's remarkable about these patterns is complete continuity of scaling here between viruses, prokaryotes, unicellular eukaryotes, and multicellular species. There's no sudden dichotomous break in the scaling of genome content with genome size in these very, very different organisms, suggesting there's some general rules for genome evolution. So I'll be talking about these general rules with respect to these major non-adaptive forces of evolution. And what turns out is every one of these scales dramatically with organismal complexity, meaning that the rules by which evolution proceeds are very, very different in unicellular versus multicellular species. This pattern here we've known from ecology for a very, very long period of time. If you double the size of an organism in any phylogenetic group, on average, it will reduce the population size by 50%. So this is a log-log plot. For those who can remember their mathematics, this means there's 20 log differences here. So that's a 1 followed by 20 zeros in different numbers of individuals per species. That means that chance plays a much greater role in us big things like vertebrates and land plants than it does in microbial species, simply because there's more flips of the coin, so to speak, out here than there is here. So this is a well-established fact from ecology in any kind of organismal group you think about. There's also a scaling between the recombination rate and genome size in different organisms. So us big multicellular species, animals again, and land plants are way down here. There's very little recombination per physical distance. and then you get into microbial species, an enormous amount of recombination. Both of these issues are big problems for natural selection. Random genetic drift plays a bigger role out here, so it blinds the eyes of natural selection, so to speak. And low rates of recombination is a real problem for selection as well. The problem here is that if two nucleotides in a chromosome are tightly linked, if a mutation arises here and it's always tied to this one, natural selection can't operate on these two genomic sites separately from each other. So you need high rates of recombination to have efficient response to natural selection. Now, the third point that I want to discuss here has to do with mutation. My lab's been working on mutation for quite some time, and now we finally can get some answers due to whole genome sequencing about the pace and the spectrum of mutations that we couldn't ever obtain before. These experiments that we do are called mutation accumulation experiments. They're probably the most boring experiments ever devised. What we do is typically we'll start with a single individual. This is a Daphnia microcrustacean that lives in all the lakes in the world, really. They're reproducing clonally. You take a single mom, we start 100 lines or so, and each generation we take one random offspring from each of these lines. We've essentially shut down natural selection. Unless a mutation is lethal, which is a very small segment of mutations, or causes complete sterility, it is trapped in these lines forevermore with a single progeny descent. At the end of the experiment, which can be anywhere from 400 to 1,000 generations, we now go in and sequence all these genomes, and everybody started out the same, and this gives us a good idea of the, as I said, the rate and the spectrum of mutations. So essentially no selection bias in these kind of experiments. You end up getting some pretty bizarre phenotypes after a short period of time, phenotypes that would never exist in nature. So this is from an experiment we did with the elegant nematode, C. elegans. That's what one of these nematodes normally looks like. And here's a rather rotund fellow that developed after a few generations due to a number of mutations. Here's a really aberrant form with a kinked tail and a protruding vulva and so on. So you can get really sick individuals because we're pampering these clonal lineages so well. We've done this work with a wide variety of different organisms now, ranging from this plant Arabidopsis to a single-celled green alga. There's a bigger view of Daphnia, Paramecium, and the nematode again, and the famous yeast, which you all profit from when you eat bread or drink beer. And we know the mutation rates from all of these, from these kind of experiments that I just mentioned. What's remarkable is there's also a scaling of the mutation rate with genome size across all of cellular life. We humans are right out here. That's the bad news. Almost all mutations are deleterious, and we humans have the highest mutation rate of any well-described organism. You can see, again, there's a log-log scaling, so there's a 100-fold difference in the per-generation mutation rate between microbes and us mammals, and there's a land plant in there. Part of this increase in the mutation rate is due simply to an increase in the number of cell divisions per generation. For a single-celled organism, one cell division is one generation, but as big multicellular things have germlines, and there are multiple germline cell divisions. So for invertebrates in this land plant, there's typically about 10 to 40 cell divisions per generation, and for us mammals, it's 50 to 400. 400 is probably an underestimate. It's probably more like 500 or 600 for old males, because there's continuous turnover in the germline of males and mammalian species. Old males are really dangerous from the standpoint of the human population. Almost all mutations in the human population come through the male side of the germline due to this increased number of germline cell divisions. So what's causing this increase in the mutation rate? As I said, it's in part due to more cell divisions and mutation being tied to replication, but not all mutations are tied to replication. And one of the most pronounced things that we now know is that our basic DNA repair pathway machinery has been incapacitated as we went from single-celled organisms to multicellular species. I'm looking at a very well-defined construct for estimating the mutation rate in various species. These are called microsatellite DNA. So the DNA will have a repeat of AT, 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 and there can be various numbers of these in different alleles. So when you hear about forensic cases, crime cases, and individuals being identified as perpetrators of the crime, these are the kind of markers that are being used. They're hypervariable markers. these kinds of loci mutate at rates. The DNA gets confused when it replicates, and they mutate at rates that increases with the number of repeats. And a nice scaling. So here's the pattern for yeast microsatellite loci, this beautiful scaling here. And dictyostelium is a slime mold that follows the same pattern. So it's going through a single cell division there. Here's data from our lab on C. elegans, the nematode. So this is a multicellular organism, about 1,000 cells, but this is the number, the mutation rate per cell division. So parallel to the yeast data, but the line is elevated, meaning there's a higher mutation rate per cell division. And here's the rate for humans and mouse and Arabidopsis, still higher. So the mutation rate per cell division is scaling 1 to 5 to 70 and going from these unicellular to mildly multicellular, very, very richly multicellular species. So our mutation rate is increased, not just because of increased number of cell divisions alone, but due to the fact that all of our DNA repair pathway machinery has been incapacitated. And the reason that's happened is because random genetic drift plays a much more powerful role in us mammals than in prokaryotes. So our average effective population size in millions is quite low compared to what it is in prokaryotes. That means it's more difficult for selection to prevent a slight mutation in a DNA repair enzyme that causes an increase in the mutation rate, or to promote, makes it more difficult to promote an advantageous repair enzyme. So there's this beautiful inverse scaling, all due to the noise of random genetic drift. One of the biggest mysteries in evolutionary genomics has to do with the evolution of gene structure. Prokaryotic genomes in bacteria typically look like this. So these are the Priuses of the gene world. They're even blue, just like my Prius. Well, they're not actually blue inside the genome. That was a joke for the non-biologist, so hopefully you caught that. So prokaryotic genes are nice, long, continuous regions of coding DNA separated by very, very small intergenic regions. They have very, very small, uncomplicated regulatory regions. And then we get into eukaryotic gene structure. I would call this the epitome of unintelligent design. There is no way that at least I could dream up a worse way to design a gene. Human genes are, and all eukaryotic genes for that matter, the coding DNA is separated by this non-coding intronic DNA. An average human gene will have about 8 to 9 of these spacer DNAs subdividing the coding DNA. We don't want this in here. Our genes are transcribed, and these pieces of messenger RNA have to be clipped out in exactly the right spot to get a productive messenger RNA. These are called exons, these are called introns. In the human genome, an average intron size is about 4,000 bases. An average exon size is only about 125 bases. So our coding DNA are little tiny islands in the sea of very harmful intronic DNA. We also have these big upstream regions on genes and big downstream regions on genes. These are called untranslated regions. They are transcribed into the message that will tell our ribosome how to make a protein. And the point that I want to make at this, we also have very, very complicated transcriptional machinery for genes that are involved in turning genes off and on. The problem with this is every one of these embellishments that you add to a eukaryotic gene makes that gene much easier to break. So if you have an intron in a protein coding gene, you have to reserve a large number of nucleotide sites to ensure proper splicing. These transcription factor binding sites are about 10 bases long. And so you have to maintain every one of those with exactly the right sequence and so on. Upstream UTRs are a problem because we have a scanning mechanism for translation initiation. If you get a mutation up here to a premature start codon for translation, you've got an unproductive gene. So all excess DNA is hazardous in a mutational sense. The cost is pretty pronounced. I'll talk about this for introns in a second. Every intron that you add is about 30 nucleotides that you have to maintain. You can't touch if you want a functional gene. Prokaryotes don't have to worry about this. Transcription factor binding sites, as I said, are about 10 bases long. 5' UTRs I won't talk any more about. It's a little bit complex. But I want to also mention this. We often think of intergenic DNA as being non-functional, having no function whatsoever. In many cases, that's actually true. And so you might think, well, that's completely innocuous, not harmful DNA. Non-functional DNA can't suffer from a loss of function mutation. However, non-functional DNA can suffer from a gain of function mutation. Intergenic DNA can pick up a mutation that will cause an adjacent gene to turn on in the wrong time or place during development, for example. So the problem with introns, I'm going to just focus on introns for the remainder of my talk as an example of this mutational hazard that we're all confronted with. A normal gene might look like this in a prokaryote, and a eukaryotic gene would have many, many of these introns. Prokaryotic and eukaryotic genes, this is the coding region, they can both suffer a mutation in the coding region that makes you make an inappropriate protein, but an intron-containing allele has an excess mutation rate to null alleles due to mutations in splice sites. The best way to get information on this actually comes from data on human genetic disorders. So here's a series of data that I put together recently. There's about 40 or 50 human genetic disorders that are known to be due to a single gene. And I'm looking here at dominant human genetic disorders or X-linked ones, so that if an offspring has the disease, you know the parents didn't have the mutant allele. So this is just like a reporter construct in a bacterial experiment. You collect progeny who exhibit the disease, you go in and you sequence the disease alleles, and you find out the source of the mutation. And so for every one of these data points applies to a different human genetic disorder. For the underlying locus, we can count up the number of coding nucleotides that are important for the protein and the number of introns subdividing those coding regions. So that's the ratio of the targets for introns to the coding nucleotide site. And on the y-axis is the ratio of the number of mutant alleles that are affecting intron splicing versus coding DNA. Turns out that the ratio of this number to that number is the mutational penalty you pay. And that is illustrated here by where these points reside with respect to these lines. So that line there is all I really want to focus on as an average. The height of that line is about 30. So that tells us that about anytime you add an intron to a gene, and keep in mind there can be dozens of introns in human genes, you've done something like adding 30 key nucleotides that we can't allow mutation to touch. Otherwise, you've got a defective allele. Well-kept secret, something you might want to worry about. I did this on the back of an envelope. It might be off by 1%, but not much more than that. About 8% of human mortality is due to mutations that occur in introns, either in our germline or in some somatic tissue causing cardiovascular disease or cancer. Absolutely no questions introns are harmful. We can look at the data on introns with respect to the population size of different organisms. And what we see is everything here over here, these are land plants and animals. Everybody has 8 to 9 introns per protein-coding gene. Now we're moving into unicellular species. Natural selection is much more efficient. Random genetic drift is a much weaker force, and introns start dropping out until you get over here where you're looking at bacteria. There are no introns whatsoever. So the data support the theory really quite well. You get more complex genomes evolving in populations of smaller size. Now, one of the problems with introns, we have historically had no idea where they come from, even though they comprise 30% of the human genome and the genomes of many other multicellular species. We've recently gained some insight into this by studying this beast, Daphnia, which is one of the popular organisms in my lab. What we've discovered in this beast is a number of different genetic loci in which some individuals have alleles containing an intron at a spot and others do not. This is how you study evolution. You need to look at mutations that they first arisen in a population and figure out what's causing them to expand or contract in population. So this is just some locus in this species. Each one of these lines in this gene genealogy represents a different allele. This little clade here at one spot here contains this intronic DNA stuck in the middle of the gene. Remarkably, there's another clade down here that has an intron in exactly the same spot, but it's not the same intron. So you can tell by the structure of this tree that it's independently evolved. And we found many, many examples of these now by sequencing multiple Daphnia genomes. Not sure why this is the case, but almost all of these intron gains are found out in Oregon. I used to live in Oregon. It's a bit of a strange place, but it's apparently even stranger for Daphnia than it is for humans. What this is saying is that there appear to be mutational hotspots for gain of this intronic embellishment in genes, in Daphnia and presumably in all other species as well. Now, one thing you can ask is, are these really harmful? I've already pointed this out with respect to human biology, but we can now ask about these newly arisen introns that are only 1,000 years old or so. Are they harmful or not? And the way you can think about that is you just go to a large number of individuals, sequence the gene, and find out how many allele copies have introns and how many don't. And what you typically find with intron-containing alleles is they're rare. They have low allele frequencies. You can compare that then to other single nucleotide substitutions in these, and it's a flatter distribution. Now, occasionally an intron allele rises to high frequency, 0.85 here, and even goes to fixation, causing a spread throughout the entire species. But this clearly is more evidence that these embellishments added to our genes are deleterious. And the reason this has happened out in Oregon is because the Oregon populations of Daphnia have historically gone through a population bottleneck. We can say something about the origin now of introns that we couldn't say before. So here are sequences of parts of a gene in a wide variety of different Daphnia. They don't have DNA in here, no intron. And here's a bunch of Daphnia clones that have an intron in that spot here. And what you'll notice here is there's a motif down in this intronic DNA here that's identical to that motif here. Here's a completely different gene that's gained an intron, and it has a motif down here that's exactly identical to that motif down there. We see this over and over and over again when we look at newly arisen introns. So what we think is happening here is that introns arise as an accident. DNA is double-stranded, there's a double-strand break, and we imagine a staggered cut. So the bottom half of this DNA is carved back here, and the upper part of this DNA is carved back. The DNA then gets filled in on bottom and top here, and that's what gives you these identical motifs here. And then something gets stuffed in the middle. Generally, that something is very, very AT-rich, and somehow the cell sees this as an intron occasionally and knows enough to splice it out. In one case, the piece of DNA that we have been able to identify is a fragment of DNA from Daphnia's mitochondrial genome, so we actually know the source. So to sum up in a few slides here, the way we're envisioning genome evolution as a non-adaptive process as this follows. We go from right to left here. We're going from species with enormous population sizes to low population sizes, long-term genetic effective sizes. This is where we are as humans. We all look at each other and we can tell we're physically different. But in terms of molecular diversity, we're the least molecularly diverse species on the planet. So what goes on here as we move from right to left is drift starts to play a more powerful role. When we get to this point here, genes can start growing these upstream and downstream leader sequences, not because they want to, but because they can't prevent this addition to gene links. Similarly, we get to this threshold here, and modular gene structure starts to evolve from a completely passive way simply because natural selection is incapable of eliminating new transcription factor binding sites. And then we get to this point here, and we can't prevent the invasion of introns. We think the ancestral eukaryote, the ancestor to all of us eukaryotes, resided somewhere around here. I want to point out that all these embellishments in the eukaryotic gene in today's organisms might look useful. If you have introns and you only have 22,000 genes, well, you could do different things with the same gene. You don't need to add different genes. You can alternatively splice an individual gene and get multiple proteins out of that gene. The problem with that, that's an argument for the utility of today's DNA. But these kind of embellishments arose well before multicellularity ever arose, so they couldn't have possibly been used for tissue-specific alternative splicing. This theory that I've developed, as Hope mentioned, you could write a whole book about this, and I believe this provides a unifying theme for many, many previously disparate observations on genome structure and the specific structure of genes, both in prokaryotes and eukaryotes. To sum up, I hope I've convinced you. I know some of you aren't from the biological sciences. Maybe almost no one is. I've presented this at a level I would normally present to my introductory evolution course. I hope the jargon hasn't been overwhelming. But what I've tried to do is point out that the population genetic environment of multicellular species is fundamentally different than it is for unicellular species. The same basic forces apply in evolution in all organisms. The relative importance of different forces is what changes dramatically. This is not to say that I don't believe in natural selection, but there's a fundamental difference between the mechanisms that get rise to changes in genome structure and gene structure and the ability of natural selection by standard processes of modification by descent to take advantage of the new form of genetic real estate that evolved in multicellular species, I think. So that's my favorite artwork of all time. And then finally, we just heard, I think from the dean, this famous quote that goes back to Dobzhansky's paper. This was actually a paper he wrote in the American Biology Teacher way back in 1973 for a good reason. And unfortunately, this is still a very useful and necessary paper for many high school biology teachers to read and so on. I actually think that the title is a bit of a stretch. You can do some pretty good biology, cancer biology, and so on without thinking a whole lot about evolution. But I'll modify this quote just slightly. If you want to study evolution, nothing makes sense except in the light of population genetics. And I've had an enormous amount of help in developing these ideas over the past 10 or 15 years, and these are just the names of various people and various projects I've been involved with. It really doesn't do service to them, but I've really learned a lot more from them over the years than they've probably learned from me. And that's one of the places I work, and we've gotten some funding to do this pretty steadily. Thank you.
Time for a few questions. Could you please use the mics when you ask questions?
Michael, you've given this talk very much from the point of view of a population or molecular geneticist. And since you used Dobzhansky, and when you were talking about your 4 factors at the start of your talk, 3 of them being non-adaptive, Dobzhansky divided evolution into 2 things. One was descent with modification, and the other was natural selection. You've shown us part of descent with modification, that is, how does variation arise? But an evolutionary biologist of a different stripe or a developmental biologist might say it's not just the genes. That genes interact with factors within the cell, with factors outside of the cell to produce variation. And since you introduced modularity kicking in at sort of the mid-level eukaryotic transition, I wonder if you could comment a little bit about those other sources of variation, which I would argue as a comparative morphologist are perhaps not equally important, but important in a very similar parallel way to what you were talking about for generating variation that natural selection looks like.
Yeah, so I really focus my talk on probably the simplest way to think about this really as a metaphor. And in fact, evolution of modularity is something I've worked at to a considerable degree, and I can refer to some papers later. And what can be shown is that modular structure of genes will arise passively. There's actually an advantage to have overlapping regulatory regions and so on, simply because it reduces the mutational target. But this is a very, very weak force, so it's very easy for modularity to arise without any direct involvement of natural selection initially. That's not to say natural selection does not take advantage of modularity later. It certainly does. But again, I'm trying to emphasize the distinction between the mechanisms that give rise to a change in gene structure and then the ability of natural selection to modify things. But, okay. So we can talk more about it later. It gets pretty technical, but I'm not disagreeing with you at all.
I'm Paul Griffiths, University of Sydney. I'm trying to understand why the logic of this argument is different from somebody who points out that the capacity of the genome to accumulate mutation doesn't have a natural selective explanation, and then uses that to argue that a very traditional kind of population genetic model of evolution doesn't really have such a big role for natural selection. That wouldn't seem to me to be more than a redescription. So if the claim is that the mechanisms that generate the variation on which selection then acts are not themselves the product of selection, that I wonder whether that really counts as downgrading the importance of natural selection. I mean, for example, if you look at the contemporary human genome, perhaps as much as 75% of of those modular genes you're talking about are functionally alternatively spliced. There's a massive amount of detailed evolutionary change going on. And the point that the fundamental mechanisms that allow that evolutionary change to happen are the result of the taking away of a certain kind of constraint on genome structure. Is that really downplaying natural selection's role in explaining the structure of the genome as we see it now, as much as the rhetoric of your talk seemed to suggest?
Yeah, so what I'm trying to emphasize is the mechanisms that led to the origin of these kinds of changes in gene structure. We're really trying to figure out the quantitative power of selection versus drift versus mutation in these cases. And what I've been trying to point out is that most of these changes we see that with respect to the origin of the structure of a eukaryotic gene can only be explained, keeping in mind they arose in a unicellular context, can only be explained by weak natural selection being overwhelmed by the power of drift. But once you've changed your basic structure of the gene, so almost all genes now have introns, well, now you've got a new form of genetic real estate. Natural selection can start doing things with genes that alternatively splice. I will argue that although there's a lot of alternative splicing going on in all genomes, and it's conceivable every gene alternatively splices, we're far from being sure how much of that is adaptive. Much of it's clearly non-adaptive, non-productive messages are being produced. And fortunately, we have some cellular mechanisms called nonsense-mediated decay that simply weed out all the background bad splicing. But the spliceosome is not perfect. So once you have introns, you're probably already alternatively splicing, not intentionally, but occasionally it might work, and then you can do something you couldn't do before.
Yeah, Ken Miller, Brown University. You talked about the increased, I think you used the word mutational hazard of a large eukaryotic gene with widely dispersed exons and splice sites and everything else, and it's absolutely true, and it's a striking feature of eukaryotic genomes compared to prokaryotic ones. But I wondered basically how well this squares up with the hypothesis that Kirshner and Gerhardt put forth in their book, The Plausibility of Life. And what they argued in there is that all eukaryotic systems have this tightly conserved group of core genetic functions that do things like replicate DNA, control the cell cycle, and so forth. And they're highly conserved. You can swap them back and forth between humans and yeast, for example, and you can get pretty good cell cycle control. You mess with those, you're in big trouble. But the idea that they advanced is outside of this set of core functions, you have an enormous, complex, and highly mutational set of genes that do change in non-adaptive ways, very much as the way that you described. And this basically promotes evolutionary complexity. In other words, it provides a target that it gives organisms the capacity of evolvability by the very act of having redundancy in genetic systems and having multiple mutational targets. And then they argue, and this is what the question I wanted to put to you, is that quality of evolvability itself, with the mutational hazard that you talked about, itself is selectable. In other words, is favored by selection. I wondered how you react to that.
Well, again, some of you might be aware that I'm probably one of the bigger critiques around of the whole concept of evolvability. The problem is it's a very ill-defined term. And I've read Gerhard Kirshner's book several times, and you won't find a definition of evolvability in their book. We have a definition in quantitative genetics. It's very explicit, something we can really measure. But I don't see anything inconsistent with what I'm saying in terms of the ability to do new things once you've modified gene structure. But I'll also say that what's turning out is a lot of the core proteins that are involved in interactions with DNA and RNA, it's turning out are actually evolving at remarkably high rates. I can't say we really understand why this is at this point, but it's turning out to be quite a surprise, and there's many different examples that have appeared in the literature just very recently. A great example being a recent paper on speciation in Drosophila that has to do with a DNA binding protein that no one would have guessed. So I probably need to get down here pretty soon, and then we can revisit it. Thanks.